The Dinka people of South Sudan are among the tallest humans ever recorded. Their average male height, measured in field surveys in the early 1950s, came in at nearly 6 feet. Their Y-chromosome lineage is one of the oldest surviving patrilineal threads in any living human population on Earth. Scientists have called their genome ancient, pristine, a living window into Africa's deep past. That story is completely true. It is also precisely half the story. Here is the half nobody tells you. In 2017, a team of researchers at Harvard Medical School needed to reconstruct the population structure of ancient Kenya. To run their model, they required 3 reference populations: a 4,500-year-old forager from a cave in Ethiopia, ancient individuals from the Chalcolithic Levant, and 1 living group chosen for a specific reason. Their genome was considered so genetically stable it could stand in for a population that had vanished from the archaeological record thousands of years ago. The living group they chose was the Dinka, not as the subject, as the instrument. Think about what that means for a second. Every major reconstruction of ancient East African history, Kenya, Tanzania, Somalia, parts of southern Africa, has relied in some structural way on the genome of a living people in South Sudan. Not because scientists were studying the Dinka, because they were using the Dinka to study everyone else. The Sudd wetlands, 130,000 square kilometers of papyrus swamp and shifting channels in the heart of South Sudan, is the geographical reason this was even possible. Egyptian armies turned back at its edges. Ottoman forces couldn't map it. British colonial surveyors failed to cross it. And for roughly 700 years, the Dinka lived inside it, genetically sealed from the population movements reshaping the rest of Northeast Africa. What the Sudd kept out was every major admixture event of the past millennium. What it kept in was one of the most extraordinary genomes in the world. If you've ever genuinely wondered where human beings come from, not the textbook version, but the real biological story, the one that's still being argued over in laboratories right now, then subscribe. Because what comes next goes deeper than most channels are willing to go. And what comes next starts with a question nobody in popular science has been willing to ask out loud. The population the Dinka have been standing in for, the ancestral lineage they're used to represent, has no published ancient genome. No name in the record. It exists only as inference. And there may be a second population, further south, carrying the same lineage with no idea what it means. About 8,000 years ago, something was moving south from the confluence of the Blue and White Nile in central Sudan. It wasn't Eurasian. It wasn't West African. It wasn't the hunter-gatherer lineage already scattered across East Africa at the time. It was something older. Something that would eventually mix with early pastoralists carrying partial Levantine ancestry and produce the people who drove cattle into Kenya 4,000 years later. And it left almost no bones that scientists have been able to find and sequence. This is not a minor gap. This is the founding lineage of the entire East African pastoral corridor. Dr. Mary Prendergast at St. Louis University spent years on this exact problem. In 2019, her team published a study in Science that tried to model where the ancestry of ancient Kenyan and Tanzanian pastoralists actually came from. To make the model work, they needed 3 source populations: a 4,500-year-old Ethiopian forager from Mota Cave, ancient individuals from the Chalcolithic Levant, and a third group to represent the deep northeastern African component, the old lineage, the one that predated the Levantine contact, the one that had been there before the cattle arrived. They used the Dinka. And here's what that paper actually says in plain language. The Dinka are not that ancestral population. They are the closest living approximation of one. The real source population, the group that seeded the corridor, that mixed with Levantine migrants, that reshaped the genetics of East Africa for thousands of years, has no name in the scientific record. It has no excavated bones. It shows up in every ancestry model as a component labeled Dinka-related, which is the field's way of saying, we know it existed, we know roughly what it looked like genetically, and we're using the closest thing we have. And yet, the data said the exact opposite of what you'd expect to find if this were just a regional story. At Kakapel Rock Shelter in northwestern Kenya, an individual buried roughly 300 years ago was excavated and sequenced. The best-fitting genetic model for that individual didn't point to nearby Luo communities. It didn't point to Bantu-speaking Kenyans. It pointed to the Dinka, a population living 600 miles north in South Sudan. The lineage had traveled. The name didn't travel with it. Science has traced the civilizations this ancestral population helped build, the cattle routes they walked, the genetic descendants they left across 5 countries. It has not named them. They appear in every paper that cites them as a source. They appear in no paper as a subject. A wave of people reshaped the genetic landscape of an entire continent and left no entry in the record. And there is one more thing they left behind, found not in any fossil, but in a 2017 statistical model that has never been described in plain language until now. The Sudd is not picturesque. It's oppressive. Dense papyrus, floating vegetation islands, channels that shift from year to year, air that sits on you. For most of recorded history, it was simply impassable. And for the Dinka, it became something that no army and no empire ever planned, an accidental fortress. While Arab conquest swept North Sudan in the 14th century, while the Bantu expansion pressed from the west, while Eurasian admixture fundamentally reshaped every population along the Nile corridor north of them, the Dinka, inside the Sudd, were left alone, and their genome shows it. In 2017, a team at the Wellcome Trust Sanger Institute in the UK, led by Dr. Deepti Gurdasani, genotyped nearly 4 million genetic markers across 221 individuals from 18 populations in Sudan and South Sudan. What they found confirmed what the isolation implied. Nilotic populations in South Sudan, Dinka, Nuer, Baria, showed near-zero Eurasian admixture. No signal from the Arab conquest that genetically transformed northern Sudan. No Bantu component from the west. The genetic isolation wasn't a theory, it was measurable. It was in the numbers. The word you'll see in every paper covering this is pristine. The Dinka genome is pristine, ancient, unchanged. Everything I just told you, hold on to it, because it's about to mean something completely different. Because the Gurdasani study found something else, something that doesn't appear in any popular account of this research. Buried in the statistical output, a test called an F3 statistic was a signal. A small but statistically significant pulse of West African ancestry in the Dinka specifically. Not in the Nuer, not in the Baria, in the Dinka alone. The value was minus 0.001038, with a z-score of minus 5.283. In population genetics, that's not noise, that's a real event. When did it happen? From whom? The paper names the direction. It does not name the event. Here's what the timeline suggests. In 1315 and 1316, the last Christian Nubian kingdom, Dongola, fell. The Gurdasani team estimated that the admixture now visible in northern Sudanese populations arrived roughly 700 years ago, consistent with that collapse. The Dinka signal is different in timing, and different in direction. It didn't come from the north, it came from the west, during the same century. The genome that the entire field calls pristine absorbed one documented, unexamined pulse of external ancestry at a specific historical moment. Science measured it, logged it as a parameter, and moved on. What the Sudd kept out was most of the ancient world. What it kept in was something that may have changed the instrument without anyone noticing. And if the direction of that ancestral flow ran the other way, if the Dinka were not just the stable baseline but an active source, then the map of African prehistory has been oriented wrong, the other way around. Let's be specific about what that means. 8,000 years ago, in the Gezira Plain, the flat, agricultural heartland between the Blue and White Nile in what is now central Sudan, a proto-Nilotic population was already genetically distinct. At some point, people related to this group mixed with early pastoralists carrying partial Levantine ancestry. The resulting population, researchers call them the Early Northeastern Pastoralists, ENP, began moving south. They reached Kenya within 4,000 years. They reached the southern tip of Africa within 6,000. They carried cattle. They carried the Dinka genetic component. And they left measurable traces of that ancestry in populations from Somalia to South Africa. The time collapse here is something you need to sit with for a moment. A person herding cattle through the Kenyan highlands 3,100 years ago, whose bones were excavated at Luxmanda in Tanzania, had their genetics reconstructed using DNA drawn from living Dinka today. 3,000 years, one genome, bridging them. Dr. Pontus Skoglund's team, then at Harvard's Reich Lab, published this in Cell in 2017. The Dinka were included in their reference panel as one of 6 populations representing major lineages present in Africa today. In the formal ancestry models, Dinka-related ancestry appeared in ancient Kenyan pastoralists at between 24 and 33%, in ancient Tanzanian individuals at significant proportions. And the best-fitting model for present-day Somali required a Dinka-related component alongside Levantine and Ethiopian sources. The ancestry attributed to Dinka in Somalia, today in 2024, is a real, measurable fraction of one of that population's founding components. This isn't a regional story. This is continental. And here's the part that stopped me cold. Manute Bol, 7 feet 7 inches, born in Gogreel, South Sudan in 1962, one of the tallest athletes to ever play professional basketball in Washington, D.C., carried in his genome the same Y-chromosome haplogroup that appears in 3,100-year-old Tanzanian ancient DNA. His patrilineal ancestor may have herded cattle at Luxmanda. Science can now establish that connection with formal statistical models. The person and the ancient bones, the genome as the bridge between them. Nobody has told that story in those terms. The standard account of Dinka history positions them as a population that received the genetics of the ancient world. The archaeogenomics record positions them as a population that transmitted the genetics of the ancient world to nearly everyone around them, and then, having done so, remained behind while the corridor moved south. And here's what nobody in the coverage of this has ever mentioned. The genetic fault line between Nilotic and non-Nilotic populations in Northeast Africa, the line the Gurdasani study mapped in 2017, correlates with extraordinary precision to a political border that has been in active armed conflict since 2011. The genome did not cause the war, but the war is being fought, in part, along the exact boundary that population genetics drew thousands of years ago. We are going to mention it. The Gurdasani team produced, among other things, a visual map of genetic distance between populations. A principal components analysis, a way of plotting populations in space according to how genetically similar or different they are. On that map, a gap appears. On one side, Nilotic populations including the Dinka. On the other, Arab admixed Sudanese populations, Beja, Nubians. The genetic divide between them is, in the study's own words, mainly caused by a large Eurasian ancestry component that arrived on the northern side roughly 700 years ago. These are not subtly different populations. In the genetic space of Northeast Africa, they cluster separately, with measurable distance between them that reflects centuries of minimal intermarriage. That line runs through the middle of Abyei. Abyei is the oil-rich region claimed by both Sudan and South Sudan. Dinka and Ngok communities have lived there for generations. The most violent episodes of the post-independence conflict, including raids, mass displacements, and direct military confrontations, occurred there. And in the Gurdasani genetic map, Abyei sits directly on the boundary between those two population clusters. Stay with me, because this next part is the reason this video exists. The standard framing of the Sudan-South Sudan conflict positions it as a post-colonial border dispute, a resource war over oil, a legacy of British administrative boundary drawing. All of that is accurate. But the genomic data adds a dimension that none of the political coverage includes. The populations in conflict are among the most genetically differentiated groups in Northeast Africa, separated by a discontinuity that formed 700 years ago and remains measurable to the present day. The border isn't arbitrary. The genetics agrees with the geography. A 2017 peer-reviewed genomics paper describes this with precision. A conflict that has killed hundreds of thousands of people and displaced millions more is being fought along coordinates that science mapped and said nothing about to anyone outside the field. But here's where it gets strange. The divergence date that sits underneath all of this, the estimate of when the ancestral Nilotic lineage split from its nearest neighbors, produces a number when cross-referenced with certain archaic admixture models that doesn't fit the current published timeline of Northeast African population history. The field hasn't built a story that accommodates it, not because the date is wrong, because the story isn't wide enough yet. Let me show you what the field does when a number won't fit. First, and I mean this seriously, let me make the strongest possible case against everything I've been building toward, because it deserves to be made and it deserves to be heard. Using a modern population as a proxy for an ancient lineage is not unusual. It's standard practice across the entire field of archaeogenomics. The San people of southern Africa are used this way, the Mbuti of the Congo Basin, the Yoruba. When ancient samples from a specific lineage are unavailable, scientists use the closest living representative they can identify, and they justify that choice with population continuity data. That's not a shortcut, that's calibrated inference. And in the Dinka case, the justification is strong. The Gurdasani 2017 study confirmed near-zero external admixture over centuries. The Y-chromosome data from Dr. Hisham Hassan's team at the University of Khartoum confirmed haplogroup stability. The PLOS genetics team explicitly wrote that nilotic populations display long-term isolation and continuity. The Skoglund Cell 2017 paper included the Dinka in its reference panel because of those confirmed findings, not despite them. The proxy isn't an assumption. It's calibrated inference, supported by multiple independent lines of evidence. These are careful scientists. The methodological framework they're working within is legitimate. What they found next changed everything they thought they knew about this. Not their competence, their scope. Because the proxy justification tests one thing: whether the Dinka genome shows significant admixture from external populations over time. It has been tested for that. The answer is mostly no, with one notable West African exception that hasn't been followed up. But there is a second question the proxy methodology doesn't address: whether the Dinka genome has changed internally, through selection pressure, through bottlenecks, through the still unexplained West African admixture event, in ways that may mean it doesn't accurately represent the ancestral lineage from 4,000 to 8,000 years ago. Using a proxy without fully characterizing that proxy is not bad science, but it is incomplete science. And here's the specific incompleteness that matters most. The Second Sudanese Civil War, 1983 to 2005, killed approximately 2 million people, the majority South Sudanese. Population geneticists have a technical term for what happens to a genome after a mass mortality event at that scale: a bottleneck. A bottleneck changes allele frequencies, and several of the studies that used Dinka DNA as a reference anchor collected their samples after that war ended. The question of whether the post-war Dinka genome accurately represents the pre-war Dinka genome, which is itself the proxy for a population 4,000 years gone, has not been formally assessed in any published study. The current model says the genome is stable. Something happened, and the model, as written, cannot see it. What the current model says didn't happen, and what we actually know. There is a database maintained jointly by Harvard Medical School and the Max Planck Institute for Evolutionary Anthropology in Leipzig. It's called the Allen Ancient DNA Resource. As of version 54, it contains genomic data from over 10,000 ancient individuals spanning Europe, Asia, the Americas, and Africa. It is the most comprehensive archive of ancient human DNA on the planet. Search it for whole genome studies where the Dinka are the primary subject. The people being investigated, the history being reconstructed, the past being recovered on their own terms. What you find is this: studies from 2017, 2019, 2020, 2022. In every one, the Dinka appear as a reference population, a proxy, a calibration anchor, not a subject. This isn't just in the Allen database. A systematic look at archaeogenomic studies published between 2017 and 2024 that include Dinka genomic data confirms the same pattern. The Dinka have been used as a reference population in at least 12 major peer-reviewed studies of African prehistory. In the same period, the Khoisan, the Yoruba, the Hadza, the Mbuti, all populations used as reference anchors, have also been the subjects of dedicated whole genome studies examining their own past on their own terms. The Dinka have not. This isn't just in your DNA right now. If your ancestry includes any East African lineage, Kenyan, Tanzanian, Somali, Sudanese, there is a real statistical probability that part of your own genome has been modeled using Dinka DNA as a reference point. You have been mapped using a compass that no one has fully calibrated. The Dinka genome has been used in more than a dozen foundational studies of African prehistory as a proxy for a vanished ancestral population, yet has never been the primary subject of a dedicated whole genome sequencing study. Science built the map of ancient Africa. It used the Dinka to build it. It has not yet turned the instrument on itself. Not because the science is corrupt, not because the researchers were negligent, because the questions weren't wide enough. And once you see that the most relied upon reference population in African archaeogenomics is also the least examined on its own terms, you cannot look at any of the maps the same way again. In 2011, South Sudan became the world's newest country. Its population, approximately 11 million people, predominantly Nilotic, became the subject of intense international attention: humanitarian, political, diplomatic. The genome of that population, which science had been quietly relying on for a decade, received no dedicated study in the decade that followed. The world's attention and science's attention were not pointed at the same thing. And deep inside the Dinka dataset, in the allele frequency distributions, in the statistical residuals of the archaic admixture models, there is a signal that has not been separately reported. Whether the Dinka carry a specific trace of a pre-sapiens lineage, different in magnitude from neighboring populations, is a question the data may already be able to answer. No one has published the answer. Here is what the data actually shows, when you look directly at it. The Dinka genome is not a pristine archive. It is a survival record. Layer by layer, it carries the record of everything the people who built it lived through, and most of those layers have never been the subject of a dedicated published analysis. The Y-chromosome lineage A1B1B2B-M13, carried by roughly 62% of Dinka men, has a time to most recent common ancestor estimated at approximately 220,000 years before the present. That number requires a moment. It means the men alive in South Sudan today share a common patrilineal ancestor with each other from a time that predates, or is contemporaneous with, the emergence of anatomically modern humans as we currently define them from the fossil record. The oldest confirmed modern human remains we have are from Jebel Irhoud in Morocco, dated to around 315,000 years ago. The lineage carried in the Dinka Y-chromosome reaches back at least as far as the species itself, possibly further. That number stopped me cold. Then there's the layer from the 14th or 15th century, the West African admixture pulse the Gurdasani study detected but never followed up on. And then the layers from the 20th century, 2 civil wars, 1955 to 1972, then 1983 to 2005. Combined, those conflicts killed millions and displaced millions more. Population geneticists understand what mass mortality events do to a genome. They create bottlenecks. Bottlenecks shift allele frequencies. They compress genetic diversity in ways that leave signatures detectable for generations. But here's where it gets strange. In 2020, Dr. Arun Durvasula and Dr. Sriram Sankararaman at UCLA published a study in Science Advances that found something remarkable in West African genomes: a signal, a trace of ancestry from a hominin lineage that diverged from the ancestors of modern humans approximately 625,000 years ago. Not Neanderthal, not Denisovan, something from Africa, something that has no name, no fossil, no face, a ghost. Their model estimated that West African populations carry between 2 and 19% ancestry from this lineage. Their dataset included non-West African populations for comparison. The Dinka data was in that analysis, used as a baseline reference. But the Dinka-specific archaic signal was not separately reported. Whether it's higher, lower, or absent relative to neighboring populations is, at present, undetermined. The data may exist. The analysis has not been published. And in 2022, researchers at the Max Planck Institute identified 3 modern African groups to use as calibration anchors for a study of ancient mating patterns stretching back 20,000 years. One of the 3 was the Dinka, chosen because their genome represented, in the researchers' words, a unique and ancient African heritage, mostly uninterrupted by later population waves. There is no ethical requirement to inform a population when their genomic data is used as a reference anchor. The requirement exists only when they are the primary subjects of the study. The Dinka were used. They were not asked. And if the post-war Dinka genome, collected after 2 million deaths, after mass displacement, after a demographic event that almost certainly created a detectable bottleneck, if that genome was used as the baseline for studies of a population that existed 4,000 years ago, then the instrument science built its models with was measuring a scar, not a baseline. There is one more layer beneath the baseline. Y-chromosome haplogroup A1B1B2B-M13, present in 62% of Dinka men, time to most recent common ancestor, approximately 220,000 years before the present. Let that scale land properly. The men who carry this lineage share a common patrilineal ancestor who was alive before the first anatomically modern human fossil we have ever found. Before Jebel Irhoud, Morocco. Before Omo Kabish, Ethiopia, at 195,000 years. The lineage predates our oldest confirmed physical evidence of our own species' existence. Based on updated phylogenetic analysis incorporating SNP and STR data, the molecular clocks of population genetics, the time to most recent common ancestor for this clade has been placed at approximately 220,000 years before present, with a 95% confidence interval of 190,000 to 250,000 years. What that means, stated plainly, is that this patrilineal lineage may have been carried by populations whose relationship to anatomically modern Homo sapiens was more complex than any currently published model fully accommodates. The field handles this with appropriate scientific caution. Popular science doesn't handle it at all. Actually, hold on. Before we go any further, I want to know something. Drop in the comments if you have East African ancestry, South Sudanese, Kenyan, Tanzanian, Somali, Ethiopian. Did you know any of this existed in your family's genetic history? And if you don't have East African ancestry, has anything in this video made you want to look into where your reference populations come from in your own DNA results? I read those comments because your answer is more connected to what I'm about to say than you might think. If your ancestry includes any East African lineage at all, Kenya, Tanzania, Somalia, Ethiopia, South Sudan, or Southern Africa, there is a calculable probability that part of your genome has been modeled using Dinka DNA as a reference point in a study you have never seen. In the formal ancestry models built by Skoglund, Prendergast, Lipson, and their colleagues, you are partly composed of the lineage this entire video has been tracing. You are a destination of the corridor that left the Jazira Plain 8,000 years ago. You arrived. You were not told where you came from. Three things are simultaneously true right now in 2024. 1. The Dinka genome is the most used living reference population in the reconstruction of ancient East African history. 2. No dedicated whole genome study has ever made the Dinka its primary subject. 3. The population carrying this genome is currently experiencing one of the most severe displacement crises on the African continent, with over 2 million people internally displaced, according to the latest UNHCR reporting. The science, the silence, and the suffering share a single GPS coordinate. If this is the kind of conversation you want to keep having, the kind that doesn't flatten the science and doesn't flatten the people, subscribe. And share this with someone who thinks they already know this story. A haplogroup estimated at 220,000 years old is alive in a man in Juba today, in a man who has lost his cattle to conflict, whose children have been displaced, whose genome is being cited in studies he has never read, and who does not need a DNA test to know who he is.